to Darbhanga Public School. Today I am going to put some light on the DNA, the genetic material. Actually I am talking about nucleic acid. Nucleic acid is a polymer, and it is two types of nucleic acid is present. One is RNA, other is DNA. DNA and RNA are the two types of nucleic acid. So if you consider nucleic acid polymer, then the monomer of the nucleic acid is a nucleotides. Means DNA, whether it is DNA or RNA, they are composed of nucleotides. What are nucleotides? Nucleotides are actually comprises of three things. One is pentose sugar, other is nitrogenous base, and the third thing which is present is a phosphate group. So these three things are going to just make up or make a nucleotide. Nucleotides are having some regions that you should have to remember, that the 3' and 5'. Whenever I say 3', it means hydroxyl group is present at the pentose sugar, and at 5' it is always phosphate group is present. Before you understand these things, first you see the nucleotide structure. In nucleotide structure, at the carbon number 1, nitrogenous base is attached. Carbon number 1 means first prime of carbon of pentose sugar. Nitrogenous bases are attached. What are these nitrogenous bases? Nitrogenous bases are of two types. One is purine, other is pyrimidines. Purines are adenine and guanine, and these are having this dicyclic rings, and there are nitrogen along with the carbon and hydrogen. Nitrogen is also present in them, and nitrogen is present at the position 1, 3, 7 and 9th position. And the attachment of the nucleotide carbon number 1 with the 9 means 1,9 attachment is seen in case of purine in nucleotide, and in case of pyrimidine, the attachment with the first 1,1 attachment means 1 carbon of the pentose sugar at position 1, and at position 1 of the this nitrogenous base, the nitrogen is present. So 1,1 attachment is seen in case of pyrimidines. If I say pyrimidines, it means cytosine, thymine and uracil. Uracil is not the part of DNA. Uracil is present in RNA. Instead of thymine, uracil is present. Now you see this nucleoside. Nucleoside, if I say this pentose sugar plus nitrogenous base, they make nucleoside. And a nucleoside, when attached with this phosphate group, then it forms nucleotide. Okay, so you see this. So if I give you some example of nucleotides, it is a thymidine monophosphate. In this, thymine is present, and you see the deoxyadenosine monophosphate. In this, adenine is present. So these are the name of nucleotides. Now you see the double helical structure of the DNA. Before that, I just want to put some light on the this DNA structure of DNA. DNA is a nucleic acid, and this was explained by Frederick Misser. He reported DNA in pus cells. DNA is an acidic substance. The term DNA given by Jacquard. Structure of the DNA, the double helical structure, that we are quite familiar with this structure. It was given by Watson and Crick. The DNA is a polymer of nucleotides that I already told you, and in the given diagram, you will see what are the position of this nitrogenous base, and you see at the carbon number 1 nitrogenous base is present, and this carbon and nitrogenous base, the bond which is formed is called the glycosidic bond. At the 5th position, this carbon is present, and to this carbon phosphate group is attached. This is a diester bond. Okay, and you will find many phosphodiester bond if you see the polynucleotide chain of DNA. So we will see all these things in the upcoming part. Before that, you see the Watson and Crick's model of DNA. What are the salient features? Normal DNA is a double helical structure. Each strand is made up of a chain of nucleotides. Okay, two strands which are running in this double helical model of DNA, they are anti-parallel. It means one strand goes from 5 prime to 3 prime. Other strand this runs in reverse direction, means other strand is 3 prime to 5 prime direction. Okay, you see the DNA model, this DNA this helix. In this you see they are actually the two strands which are running parallel to each other. They are just twisted ladder type structure, and they look like a twisted ladder structure. And I'm talking about a B type of DNA. There are 5 types of DNA. I'm talking about the B type of DNA, which is right-handed twisted this structure, and this diameter of the helix is 20 angstrom, and the helix completes its one turn, that is 360 degree. The total distance which is covered in one turn is 34 angstrom. And you see in this the DNA structure, there are the grooves which are formed. One is the shallow groove, and other groove which is formed is the deeper groove. So if you have to just find what is one turn is deeper groove. When you move from one deeper groove to deeper groove, you will find one turn. Otherwise, you can also go with this shallow groove to shallow groove, then also you will find one groove. Now you see after that this polynucleotide structure is shown to you. In this you see how this arrangement is there, how the placement of the nitrogenous bases are present, and you see the in between the two consecutive nucleotides in the polynucleotide chain there is a phosphodiester bond. Bond is present, and this and glycosidic bond is present in between this carbon number 1 to the nitrogenous bases. Okay, now you see and the picture of the DNA is obtained by this X-ray diffraction technique. So you see, and here in this DNA structure you will find that the base pairing which is seen, it is A is always going to pair with the thymine, adenine and thymine pairing is seen, and cytosine and guanine pairing is seen. So in between adenine and thymine, double hydrogen bond is there. In between this cytosine and guanine, there is a triple bond. Okay, Chargaff has given a rule that the amount of adenine must be equal to the amount of thymine. So A upon T, the ratio is comes to be 1. Like the same in the ratio of C and G also comes to be the same. Now what are the types of DNA? You see the type of DNA is decided on what basis? The basis on the basis of function. If you divide the DNA, it is of two types: trophic DNA and genetic DNA. Trophic DNA, you see that it is responsible for all the other metabolic characters. Metabolic activities are controlled. Then it is trophic DNA, and genetic DNA is solely responsible for the reproduction process. Okay, if you see the paramecium, in paramecium two nuclei are this nucleus is present, macronucleus and micronucleus. Macronucleus is actually a trophic DNA because it is responsible for that metabolic activity. It controls metabolic activity, whereas in this the other type, micronucleus, it is the nucleus which is having the genetic DNA. Okay, after that you see the various types of DNA that I told you is the 5 types of DNA on the basis of number of base pairs do they have. In A type 11 base pairs, B type 10 base pairs, C type 9.33 base pairs, D type it is 8 base pairs, Z type it is 12 base pairs, and coiling A type right-handed coiling, B type right-handed, C type right-handed, D type right-handed, Z type left-handed coiling is seen. Okay, so this is and a polynucleotide chain is already I discussed. So you can see the polynucleotide chain, and in this polynucleotide chain you will find the phosphodiester bond, and how these phosphodiester bonds are going to link up with the other nucleotide and give a complete sequence. Remember that whenever there is a 5 prime, it always tells or indicates the phosphate group, and 3 prime then it indicates the OH group. What is the difference between RNA and DNA? In RNA at position number this 2 there is a hydroxyl group, but in DNA at position number 2 of pentose sugar there is hydrogen, so that is why it is deoxyribose. And RNA is this unstable due to the presence of 2 functional group at position number 2 and 3, and in DNA it is due to the absence of hydroxyl group, it is quite stable. So DNA is the genetic material of almost all living system except some viruses and bacteriophages. You will find DNA in each and everything. And what is the this arrangement of this packing of DNA? DNA is packed inside the chromosomes by some compact structures and beaded structures. They are nucleosome. In the upcoming video, I just told you about what are these nucleosomes and how this binding of the DNA take place. Thank you.